we were motivated to do this study by a very obvious fact that anyone who's seen a baby can appreciate, and that is that young mammals, and humans especially, behave very differently than adults. We had some hints from work that we and others had done in late stages of the development of muscle that there is a certain amount of extra wires, like double the number of wires one sees in the adult in young animals at about postnatal day 7 in a mouse. And that work had been published quite a while ago, and we were wondering whether that's the extent of the extra wiring, or if we looked earlier in development, might we see an even more profound change? We really didn't know, and we couldn't answer this question because technically we didn't have the tools to allow us to look. Things get very small and compact, and it's just very hard to resolve single inputs. But J.D. Wiley and Juan Carlos set about to ask two basic questions about the development of the nervous system at birth, which is, in a distinction to the adult where there's one input per muscle fiber, how many different axons converge on a muscle fiber in development? And the second question is, whereas in the adult an axon might innervate 5 or 10% of the muscle fibers, what percent of the muscle fibers does an axon innervate at young ages? And I'll let Juan Carlos tell you about how they did these experiments.
We went on and set up these experiments to find out basically how big these axons can be. And for that, we started to use animals that were developed in the lab. These animals express the green fluorescent protein in just a single axon. So using these particular animals, we went on to look at different stages of the development of the animals. So we started to look at the embryonic level all the way to adulthood. And we found, by looking at these axons, that just at birth, right when the animals are born, these axons innervate pretty much all the muscle fibers that are present. That means that every single axon touches or make connections to pretty much all the target cells that it encounters. So we were able to see a single axon, but we wanted to know too how many axons are doing the same into the target cells, right? Because as I say earlier, we were able to just look at a single axons, but we wanted to know how many of the other axons are there that we couldn't see. So in order to do that, we use a serial electron microscopy and reconstruct an entire connection with all the axons that are innervated in a single cell. And we found that around 10 to 11 axons are innervated in each single muscle fiber. And if we look at other targets, we find roughly the same number. So suggesting that pretty much all the axons that are innervating these muscles are doing the same. So they are highly divergent, and by this I mean that they go to all the muscle fibers, right? So they make connections with them, and at the same time they are highly convergent. So they go to a single muscle fibers, and they all make connections with that target cell. And this seems to be a very characteristic feature of the peripheral nervous system. As Jeff was saying, it could be also a very important feature of the central nervous system. This is obviously something that remains to be discovered or demonstrated.
Yeah, the interesting question might be, does a baby start out with a brain filled with an infinite number of possibilities of what it might become, and then experience going to school, listening to its parents, works its way on the nervous system and knocks out the vast majority of these possibilities, and you end up being, the narrow-minded adults that people like me become, at least according to our children who still have open minds. And that may be because the reason I'm narrow-minded and believe synapse elimination explains all this may be because I've lost the ability to think of other alternatives because I've trimmed away those connections in my brain.